Good morning, Miss Bader. Here is my Unit 3 visual model project. I did targets 1, 4, and 5, and I'm here to answer them for what molecules work to pass on traits from one generation to the next, how is genetic variation created through natural processes in sexual organisms, and how is biotechnology used to better understand and improve lives of organisms, including humans. So here we are going to be getting started on number 1, or what molecules work to pass on traits from one generation to the next. Alright, getting started here, we are going to read what molecules work to pass on traits from one generation to the next. So zooming here on the top left, we are going to get started. The molecule that is responsible for holding, handling, and passing on traits from one generation to the next is DNA. Here we're going to ask about what is DNA, how does DNA work, and what is DNA's job? How does it do whatever it does? What is it? DNA stands for deoxyribonucleic acid and is the genetic information inside of the cells. It's what makes us us and what makes us capable of anything. DNA is the how-to book passed on down through generation to generation. DNA holds all the plans and instructions for development and functioning of living organisms. Found in the nucleus of most cells, holding the more commonly referred to blueprint of life. But how we all have this blueprint is due to inheritance. Down here in the middle, due to inheritance during reproduction, DNA is passed on or gifted by an individual's parents to their offspring. This gift doesn't only give DNA, but inside the DNA, the even smaller molecules that make up DNA, called genes, that hold traits, are also given to both from the parents to the offspring. Traits from both parents or even a single parent can be passed down from generation to generation. Here I have written both types of reproduction and how they both work to pass down the genes. So from sexual reproduction, you have cross-selection and the mixing of genes or DNA that both gives life and creates the shuffling of the two different types of DNAs. And asexual reproduction that simply makes a genetic one-to-one of the single parent, keeping the same DNA genes rather than inheriting different traits, making a copy. So moving on to the right, we are going to ask, what does DNA look like? How does DNA function? And what holds the DNA strands together? What makes all of these up together? So getting started right at the top, we are going to talk about what holds the DNA together. Starting here with hydrogen bonds. These are the bonds placed in between each adjacent strands of their base pairs. And what are these base pairs? Base pairs are the pairs of nitrogenous bases that connect inside of DNA. These are what make up genes, and these are also what can be messed up or incompleted and cause mutations. There are 4 bases that make up these helixes. So here below I have written the 4: adenine, thymine, guanine, and cytosine. I've also color-coded it so you can see which go to which, as cytosine goes to guanine and adenine goes to thymine. Here we're going to ask what DNA is. What does it look like? DNA is formed in a double helix displayed here. This helix is made up of the sugar phosphate backbone, the base. These polymers of nucleotides that home the bases in their bonds comprise of 3 components, a sugar group, a phosphate group, and a base. As shown here, the backbone are the red and blue helixes that go down. These are what hold the bases and their bonds. So at the very bottom, all together, DNA and the genes that comprise the DNA are what pass on the information and instructions to survive and reproduce. However, genes are what give traits from one generation to the next. This can be done by one parent or 2, as both types of reproduction can pass on DNA and the parent's traits. But there is more to how the body uses DNA. Here, such as with RNA and mRNA, or messenger RNA, that's like the envelope of the instructions that get sent throughout the body. So DNA is responsible for all the instructions given. This can be whether to repair injuries or even just for growing up. Alright, on to my second target. Number 4, how is genetic variation created through natural processes in sexual organisms? Here's a more lengthy one, but I'm going to try to speed it up. Genetic variation is the difference in a species or their genetic makeup within different or the same species or populations. So here we have 3 main contributing factors I'm going to try to hastily go through. So starting here at the first one, mutations and abbreviations or changes in things. Here below I've drawn little diagrams of a normal gene and the protein that the normal genes produce, and a mutated gene with an inefficient or wrong nitrogenous base that we talked about before. These can cause either no protein or a dysfunctional protein as written here, this little red blob. Here on the left too, I also wrote the matching nitrogenous bases and a mutation with the circled is a wrong base, which is going to cause these mutations. Mutations, as earlier stated, are changes, and here's a closer look in the diagram above. Then the DNA strands, you can see the nitrogenous bases in their sets of 3 codons, but one of the bases isn't the correct pair, leading further down the line to more consequences or the mutation. Above and to the left, we can see a gene mutation and the mutation's immediate effects, like the creation of the faulty protein or no protein at all. Although fundamental for evolution, these faulty cells can be quite dangerous and even fatal in some cases. Below, I have drawn a little radiation symbol, and, surprisingly, other than generations or sudden random occurrences in the DNA, RNA, or RNA creation/replication, radiation is another known induction of genetic change. This is also an environmental factor, but what do mutations lead to? Here I've drawn a little bit of what they can lead to, being genetic disorders, others being cancer, some being nothing at all or going unnoticed in some species or populations, or even benefits. Some species or populations may see benefit from random changes, or these changes, mutations, can be good, giving some, like giraffes, longer necks for versatility or to gain food. But most importantly, what can come from mutations is how common they can be. Mutations can become a trait that stays in the next populations or generations to come. So on to the next one for how these differences occur. On to gene flow, or the mixing / giving of alleles. So here I've drawn on top an island of only white feathered birds, which eventually will have migrations of other birds. So in the middle, thanks to the isolation, other factors such as migration or isolation, these birds travel, bringing their genes to new territories, creating new variation among the bird populations already living there. This can be a bottleneck effect, which I will explain in a second. These birds that inherit the island are only blue, meaning this bottleneck effect that caused these birds to trade such merged traits are shown below. So here you can see the once all white birds have had some genes mixed with the blue ones that migrated over. As shown below, over time, this variation and become more mixed as the generations of birds developed. Different color wings or feathers. Some of them even see beak changes, or depending on the food in their region, while other birds have gained or lost size due to these same variations. But all of these have came from some sort of evolution, thanks to the migration of the birds. Gene flow is the movement of genes between populations. This can introduce new or more variations to populations, especially through migration or other means like isolation. Gene flow is just the movement and passing of genes and traits from one to the next. Moving on, we are going to get into recombination, recombine. So here I've drawn a little diagram of crossing over. Here the homologous pair moves closer together, the chromatids, and they may exchange genes. Genes that have crossed over and combined, here as shown right below the second. Recombination shuffles genetic material as shown above, leading to new combinations of traits and offspring and increasing the gene's variation thanks to the mixing of these two homologous chromosomes. Crossing over results in new combinations of genes and their traits and alleles. Meiosis is a good example showing how the cell division process creates, crosses over, and then divides into 4 different shuffled daughter cells. Here is the more definition. Recombination is a mechanism principle of when variation is introduced to populations. Example, during the mitosis or meiosis, where the homologous chromosomes, one derived from the mother and one derived from the father, merge and then split into, like I've shown above, the 4 different daughter cells. But what is the importance of all these mechanisms and gene variations, all of these traits to aid in evolution and survivability in species or populations? Not only that, but how do over time species mix and change, whether due to mutation, crossing over, or environmental pressures that cause change within these species. These are natural processes in which create, promote genetic variation. Written below here, I have a little more specific on what each are. So zooming in, we have genetic isolation, which is where little or no gene flow exists. So this can be between populations due to factors like geographic separation or reproductive barriers. These can be man-made. So these man-made factors can be stuff like artificial selection that we do, which can be the selection of preferable traits in a species that we would want. These are the man-made factors that can genetically isolate or even genetically alter certain animals and their traits. Moving on to the next one, at the very bottom here, we have geographic isolation. So here's the more physical, non-man-made. So things like mountains, oceans, or rivers that can entirely stop interbreeding or leading to genetic divergence and potentially extinction or the possibility of new species due to this inconvenience. On to my third and final target, question number 5. How is biotechnology used to understand and improve lives of organisms, including humans? Biotechnology has been used in various ways to track, monitor, train, regenerate, and even alter some genes or traits. Most of our biotechnology, however, is agricultural. As humans, we modify everything, but plants have always been both crossbred or mutated in ways that benefit or use to give preferable traits or to even take or mitigate harmful traits. Some of our most important data is from biotechnology, whether this be monitoring or being able to study things we might not have been able to study before, or even to study mutations from either birth or not. Not all genetic engineering has been perfect. As for with the modern day turkeys not being able to be inseminated by themselves, have to be artificially done by us now, but even to our man-made salmon that were causing all of the other salmon to fall greatly behind, or being able to reproduce and just having weaker offspring or smaller offspring as the years progress. But without these, however, with our work, variation in many species and crops has skyrocketed, specifying more and more capabilities in our medicines and treatments for these genetic disorders or mutations. For those being like some of the best research and biotechnology we have used for today to help us as humans is to counter cancer and sickle cell sicknesses. Here below I have drawn a little diagram This is more marine biotechnology, but still biotechnology. The biotechnology between the discovery and research on the blue whales have been incredible, being that we can now see and understand the whale singings and noise being a mating call that we had no idea about. So with that said, that is all my pleasure. Thank you for your time, and